Cell movement is a vital process in the function of all organisms. There are many varying processes which rely on cell movement, such as the development of an embryo, the movement of cells to heal wounds and respond to the immune system. Cell movement can also be seen in cancer metastasis and sperm motility. Chemotaxis is the term used to describe the movement of cells towards a favourable environment by chemoattractants, or the movement of cells away from unfavourable environments by chemorepellents. It is essential for the cells to move to areas where they can carry out their specific functions, as not all cell types are found in all areas of the body. There are two types of cell movement: cell crawling and cell swimming. First, cell crawling. The contractile units in non-muscle cells are similar to muscle cells in the way that they are composed of actin and myosin. The cell contractions are limited to a thin layer underneath the plasma membrane called the cortex. Actin and myosin too are responsible for contraction by sliding the actin filaments over the myosin, which resembles the sliding filament model which is observed in muscle contraction. Contraction is achieved by phosphorylating one of the myosin regulatory light chains. This promotes the assembly of myosin as well as increasing the catalytic activity, enabling contraction. This reaction is catalyzed by myosin light chain kinase, also known as MLCK, which is regulated by calmodulin. Increases in cytosolic calcium causes calmodulin to bind to the kinase, which results in the phosphorylation of MLCK. A myosin light chain kinase complex is formed, which activates the myosin, allowing contraction to take place. The process of cell crawling can be broken down into four main stages. 1. Leading edge extension and protrusion. 2. Substratum attachment and adhesion of leading edge. 3. Trailing edge retraction and deadhesion. And 4. Tension at front and rear of cells generates contractile force. The movement of cells through tissues is primarily due to the polymerization and depolymerization of actin filaments. A stimulus, such as a chemoattractant, activates ARP23 protein complexes. These then undergo conformation changes to resemble the barbed end of an actin filament, allowing free ATP actin filaments to adhere. This results in the nucleation of actin filaments. This process is promoted by profilin molecules. Once new actin filaments have formed, ARP23 binds to the side to create branches. Actin continues to bind and polymerize at these points. The barbed ends are blocked by capping proteins, as can be seen in the diagram. The barbed ends of the new filament causes a projection called lamellipodium, which extends towards an attractive stimulus or away from a repellent stimulus. The actin depolymerizes at the pointed ends of the filaments at the rear of the cell. This process is promoted by cofilin. Cofilin binds to the actin ADP subunits, allowing actin to be released. Actin monomers are recycled back into filaments at the leading edge. This creates a treadmill effect by polymerizing at the barbed end and depolymerizing at the pointed end. Actin is present in high concentrations in the lamellipodium, whereas myosin is found just behind the lamellipodium. The rest of the cell is pulled forward due to traction forces generated by focal adhesion sites on the bottom of the cell. The greatest traction forces occur just behind the leading edge. Cell crawling can be seen in many different functional processes, including the development of an embryo, the immune system, in wound healing, and also in cancer metastasis. First, in embryology. The body is made up of many cells which are anatomically and physiologically different to one another. This is due to the differentiation of different cell types in the body during the development of an embryo, called morphogenesis. These cells migrate and change shape to become particular cell types, for example, blood cells, or ciliated cells in the lining of the lungs. Gastrulation is the event during which 3 germ layers of the embryo are formed: the ectoderm, mesoderm, and endoderm. This formation of the layers happens by movement of the epiblast cells, creating 3 distinct layers. Following this, primary neurulation occurs. This is the phenomenon by which ectoderm cells fold inwards to form the neural tube, which is the precursor of the central nervous system. The initial change in cell shape causes the square ectodermal cells to morph into rectangular cells by the elongation and reorientation of microtubules inside each cell, causing them to become long parallel tubules. These cells are located in the mid-dorsal region of the embryo and form the neural plate. This plate then begins to fold as the outer extremity cells of the neural plate change their shape by contracting actin filaments and forming wedge-shaped cells. These wedge-shaped cells make the entire neural plate contract and curve in on itself. Eventually, the outer extremities will touch each other in the centre, forming a hollow tube, which will eventually give rise to the entire nervous system. This type of cell movement in embryology is one of the most key events to take place during development. If the cell movement does not occur, it can have very serious implications in the function of an organism, seen in neural tube defects. The most common neural tube defects are spina bifida and anencephaly. These open tube defects occur when the brain or spinal cord are exposed at birth. Anencephaly. In this defect, babies are born without parts of the brain and skull. This happens because the neural tube forms, but then doesn't close all the way in the upper part. This then results in the babies having no forebrain or cerebrum, with the remaining brain parts there but not being covered by bone or skin. There is no cure and almost all babies will die shortly after birth. Spina bifida. This is a birth defect that affects any part of the spine due to the neural tube not closing all the way. This means that the spinal cord and nerves that would normally be protected by the backbone are often damaged. This defect is variable in severity. Some sufferers have no motility, while some may have mostly normal function. Another form of cell movement can be seen in the immune system. Cell movement is a crucial component of a functioning immune system. There are several instances where leukocytes may need to migrate to different tissues, but an area of particular importance is during inflammation, where the body attempts to defend itself against pathogens. During this, leukocytes present in the blood move into the area of infection using a process known as extravasation. Upon interaction with a pathogen, resident macrophages and mast cells in the tissues release cytokines and other chemicals that induce inflammation. These signalling molecules affect the endothelial cells of the blood vessels surrounding the tissues and cause them to produce and present a group of molecules known as selectins on their surface. Selectins are glycoproteins that function as cell adhesion molecules. L-selectins are found on the surface of most leukocytes. E-selectins and P-selectins will be found on the surface of endothelial cells after being activated by cytokines. Leukocyte extravasation can be broken down into 4 main stages: capture, rolling, arrest, and diapedesis. The first, capture, occurs during inflammation after E and P selectins are presented on the surface of the endothelial cells. This allows ligands, such as P-selectin glycoprotein ligand 1, that are found on free-flowing leukocytes, to bind to them, triggering the next steps. However, for leukocytes to be able to do this, they must first be recruited to the area, which is done through the use of chemokines, a type of cytokine that induces chemotaxis. During inflammation, these act to guide leukocytes to the region of infection. Once the chemokines have been presented on the surface of the endothelial cells, the second stage, rolling, is initiated, which acts to slow down the leukocytes. The selectins here bind transiently to their ligands, so the leukocyte doesn't stop completely, but starts to roll along the endothelial surface as more selectin-ligand interactions occur, until it slows down. The rolling also triggers the activation of another group of proteins known as integrins. A class of these, known as beta-2 integrins, are found on the outside of leukocytes and correspond to a set of receptors present on endothelial cells. The selectin and chemokine binding causes a conformational change in the protein from closed to open, which leads to the third stage, arrest. Once the beta-2 integrin molecules are in their open conformation, they are able to bind to ligands on the endothelium, such as intercellular adhesion molecule 1, which then tethers the leukocyte and stops it from rolling. After arrest, the leukocyte cytoskeleton will undergo structural changes that allow it to spread and crawl along the endothelial cells, directed by chemokines, until it reaches a junction. Diapedesis can then occur, which is the movement of blood cells through capillary walls. So the cell passes through the endothelium and into the tissue. This transmigration can either be paracellular, which is between cells, or transcellular, which is through them. Mutations in the genes associated with integrins and selectins can lead to leukocyte adhesion deficiency disorders, abbreviated to LAD. As an example, LAD1 disorder is rare, which can affect a specific beta-2 integrin. As the receptor isn't assembled properly, it means that neutrophils can't bind to or move through the endothelial cells. The result of this is that individuals with this disorder have recurrent bacterial infections, which can be fatal. Neutrophils play an important role in the body's defence against foreign invaders. Now moving on to wound healing. The skin is an important organ of the body as it provides one of the first lines of defence against infection. This layer of cells provides chemical and physical protection against potential pathogens. If this barrier is breached, then the body must repair itself or risk serious consequences. In order for wound healing to take place, several cells such as neutrophils and macrophages must move to the site of the wound to kill any microorganisms that may cause infection. In addition to this, fibroblasts must move to the site of trauma to remodel the damaged tissue. Fibroblasts move to the site of the wound via cell crawling through the tissue. They are attracted to the site of infection by a paracrine chemoattractant secreted by leukocytes. After the coagulation cascade has occurred and a fibrin clot has formed, fibroblasts start to infiltrate the wound and dissolve the fibrin clot, replacing it with a collagen matrix. In the final phase of wound healing, the composition of extracellular matrix is changed over a period of months, again by dermal fibroblasts. Fibroblasts move by projecting filopodia and lamellipodia. However, they are not attached to the substratum by focal adhesions, so the leading edges of the cell tend to be seen as ruffles. If cell movement did not take place, wound healing would not occur and infection couldn't be prevented. Moving on to the final example of cell crawling, cancer metastasis.
Cancer is unlike the other methods of cell movement that we are looking at, in that it's not a lack of cell movement that causes the disease symptoms. Instead, it is the cells moving to another place in the body that causes the cancer to spread and the symptoms to worsen. Cancer cells that spread to a distal organ of the body and induce tumour growth lead to what is known as metastatic cancer. There are various ways in which cancer cells move. Collective cell migration allows the cells to move as a sheet due to cadherin proteins at gap junctions. This is seen in melanoma and epithelial cancer cells. When the cadherin junction protein is suppressed, the cells can separate by dedifferentiation from each other, leading to individual migration away from the primary tumour. There are two types. Firstly, there is mesenchymal cell migration. Integrin-dependent interactions adhere the cell to the substrate. Proteases are released which remodel the extracellular matrix into a path which the cells can follow. This is seen in elongated cancer cells like gliomas and fibrosarcomas. Secondly is amoeboid cell migration. Actin and myosin contractile forces in the extracellular matrix squeeze the cell body, allowing cell motility. There is no degradation of the extracellular matrix as the cell shape changes to fit through the gaps already available. Adhesion to the substrate in this method is integrin independent, making it much weaker. This is seen in cells with a rounded morphology like lymphoma and small cell lung carcinoma. Cells can change between the two individual types, but they can't differentiate back to the collective migration. When the cadherin junction protein is suppressed, cell-to-cell adhesions are lost, allowing the cell to separate from the collective sheet. The epithelial to mesenchymal transition, or EMT, forms individual cells that undertake mesenchymal migration. Then, if integrin and protease functions are weakened in these mesenchymal migrating cells, there is the transition to rounded cell morphology and amoeboid movement known as the mesenchymal amoeboid transition, or MAT. Both cell-to-substrate and cell-to-cell adhesions are inhibited, but as shown by the double arrow, this transition can reverse if the environment changes. Cells in the collective sheet can also transition straight to amoeboid migration by the collective amoeboid transition, or CAT, pathway. When the cells are able to move, they can disseminate from the primary tumour. Cells from the primary tumour progressively grow with nutrients from the host. Proliferation continues and a vascular network is established to the tumour. Cells detach from the main tumour, and after they have invaded adjacent tissues, they penetrate through the endothelium of lymphatic or blood vessels to enter systemic circulation. This process is known as intravasation, and only a few cells survive through circulation to the next step. Surviving cells adhere to vessel walls in the capillary beds of distant organs. Here, they are extravasated through capillary endothelium and move into tissues. In the tissue, the cells proliferate from small micrometastases into metastatic tumours. Metastatic cancers resemble the primary cancer from which they came from, in that the cell type is the same. When we look at a breast cell, it is a primary tumour if the cell type is normally found in the area, so breast cells in a breast tumour. But if the cell type does not belong there, breast cells in a lung tumour, then the tumour is a metastatic tumour.
In addition to cell crawling, another form of cell movement is cell swimming, usually controlled by the movement of a tail-like flagellum. This type of cell movement varies depending on the nature of the cell or the organism. Two examples have been given here: sperm motility and bacterial movement. The motility of the sperm flagellum is driven by the cytoskeletal elements in the sperm tail known as axonemes. Axonemes are composed of dynein, their regulatory bodies, and also the 9+2 microtubules. The movement of the flagellum involves microtubule filaments sliding along one another. Dynein arms are the generators for the force that slides the filaments within the axonemes. The arms on the subunit A tubule of one of the doublets uses a walking action to move towards the base of an adjacent doublet, subunit B. The subsequent generation and breakdown of cross bridges between the tubule and the dynein arm requires ATP. This sliding translates into bending when there is a section of the axoneme which resists the sliding forces. Because of the way the 9+2 structure is arranged, when half of the microtubules slide along each other, they cause a bend in one direction, and the other half generate a bend in the other direction. Deficiency in the axoneme structure leads to a reduction in sperm motility and ultimately a higher chance of infertility. Asthenozoospermia is the term used to describe reduced sperm motility. It can be caused by a primary ciliary dyskinesia, which is where there are structural abnormalities in the sperm flagellum. This disease is genetically inherited, and the consequence of the mutation is that the components which make up the flagellum, such as the central apparatus, inner and outer dynein arms, and radial spokes, are non-present or non-functional. This means that the entire axoneme structure is also non-functional and so lacks the ability to move. Asthenozoospermia can also be caused by mitochondrial DNA mutations in human sperm. Sperm cells require a huge amount of ATP in order for the motility of the flagellum to be maintained, especially in the early stages of fertilization. Therefore, a lack of mitochondria can have serious effects on the mobility of sperm.
A number of bacteria swim through the use of flagellum, as seen in E. coli for example. They come in four main forms: monotricious, ampitricious, lophotricious, and perititricious. These names refer to the number and arrangement of the flagella on a single microbe. Monotricious bacteria have only a single polar flagellum on one of their ends. Ampitricious are similar, however they have a flagellum at each end. Lophotricious and perititricious are both types of bacteria with multiple flagella. Lophotricious have a bundle of flagella at one end, whereas perititricious have the flagella spread out all over the bacterial cell. The structure of a flagellum is key to understanding how certain bacteria are motile, and as such, how they can be effective at causing disease. We'll start with where the power for movement is generated, conveniently named the flagella motor. It consists of two main components, a rotor and a stator. The rotor is comprised mainly of a protein called flig. This is bound to the MS ring non-covalently. The stator consists of two main proteins, MOT-A and MOT-B, and is bound to the peptidoglycan. It is believed that the charged residues of MOT-A and the carboxyl terminal of the flig protein are responsible for the generation of torque. This is all powered by the formation of a protein gradient. A current theory is that when the protons pass through the membranes, they bind to the stator, causing a conformational change. This in turn drives the rotor. The basal body is the other key structure of the flagellum to look at. It is anchored in the cytoplasmic membrane of the bacteria and consists of four different sections. These are the LPS membrane, peptidoglycan, motor switch, and a rod which transverses the membrane. The role of this structure is to transfer power from the motor to the other structures of the flagellum, principally the hook. From there, the power is used to move the filament in order to generate motion.
This concludes the overview of cell movement and some of its implications for function and disease. For further information, please consult the link to the website in the description below.